New COVID vaccine design offers cheap and effective product for more of the world. Uneven global vaccine coverage against COVID-19 still persists today. It risks of new variants because the coronavirus can conveniently jump between the vaccinated and unvaccinated. At MIT, a new vaccine approach may help bring the solution to more parts of the world. The most effective COVID-19 vaccines use mRNA technology. Known for their innovative approach, mRNA vaccines can easily be adjusted to engage new threats. However, the tech has big downsides that made it impractical for many territories. One downside is the need for ultra-cold storage, something rare in very poor regions. To help even out vaccine coverage, scientists at MIT and Beth Israel Deaconess Medical Center are developing a new vaccine. They said that it is an inexpensive, easy-to-store, and effective alternative to RNA vaccines. Published in the journal Science Advances, scientists revealed that the vaccine is made of fragments of the SARS-CoV-2 spike protein arrayed on a virus-like particle. Early testing suggested that the technique produced a strong immune response and protected animals from the viral challenge of infection. The entire goal of this new vaccine is to offer something effective and easy to manufacture. The team ended up with protein subunit vaccines, products that contain small pieces of viral proteins. The tech behind it is recognized and has yielded vaccines against hepatitis B and acellular pertussis. Earlier in this pandemic, a basic subunit vaccine wouldn't be useful because the small piece of the virus's spike protein can't produce a strong immune response on its own. To up its performance, the team went for a full display approach, a virus-like particle showing several copies of that fragment. For this study, the team selected the hepatitis B surface antigen as the scaffold. Then, it was coated with the SARS-CoV-2 fragments. When tested, the coated particle triggered a better immune response compared to the fragments alone. Yet there's still the challenge for easy, efficient, and cheap manufacturing. The team optimized the vaccine design to accommodate an engineered yeast Pichia pastoris. They confirmed that the protein fragment and hepatitis B particle can be produced separately in yeast. In non-human primates, the team combined the vaccine with adjuvants already used in other vaccines. Their vaccine demonstrated antibody levels similar to some existing COVID vaccines, like Johnson & Johnson. Vaccinated animals also showed lower viral loads after exposure to the coronavirus, compared to unvaccinated animals. On top of practical features, their vaccine is modular. It can be tweaked to fight new variants using protein fragments from those. Imagine the vaccine as a plug-and-play weapon against this pandemic. What do you think of producing vaccines using yeast? Which do you prefer, optimize or create new COVID vaccines? Share your thoughts in the comment section. Till our next video, thanks for watching.